get started. You guys know the game by now. You should be lining up at the microphones with all the wonderful questions. But while people are pulling their notes together here, let's go ahead and take our first online question, please.
Okay, I've got one here. Can the heat effect be proven by in vitro test, or do we need to prove vivo?
As I shown in my presentation, we have looked into and developed an in vitro approach based on IVPT study. And yes, you can evaluate heat effect of your test TDS product and compare with the ROD using IVPT.
Okay. Microphone number one, nice close for me, please.
Hello, this is a question for Dr. Bennett. So the current situation for the TDS impurity systems is the limit for impurity is still following the limit of the oral products. So what is the agency's opinion on the impurities showing no systematic absorption or lower absorption than active drugs?
Sorry, I'm not sure I understand the question.
So I think the question is mainly focusing on, so the TDS impurity limits is currently following the same as the oral product limits. So what is the agency's opinion on the impurity levels if the active drug is not showing any systematic absorption or any lower absorptions?
So as far as impurities, it sounds like you're referring to drug substance related impurities. So yeah, so it followed the ICH guidelines for that, but we do base, it's important to understand that we base the MDD and we calculate the ICH levels based on the total drug load in the system and not based on the nominal delivery rate of the drug. So basically, it's not treated the same as orals, and oftentimes the thresholds for impurities are lower than they would be for to your point about I guess I still don't understand the question.
I think you're.
And for the sake of time, but during the next break, which will be lunch, if you two can dig into that a little bit deeper, would that work?
Yes, sure.
Sorry,
Thank you.
sure.
Thanks.
Sorry.
Okay. All right. Nice close, please.
Hi, my name is Shu Wang, working for the Biomedics. Okay. The question is for the Dr. Bennett. You mentioned about that the transdermal release paper take the API out, and that's probably due to the tight release issue. And before, I used silicon-29 NMR to study both liquid and solid to study the tight release and easy release. And I understand you are NMR expert. Okay, so in case we cannot adjust the formulation to make the release correctly, what's the choice? Go for the easy release, so probably peel out too loose, too early, or choose the tight release?
From my own perspective, neither would be acceptable.
Neither? Okay. Okay, great, thank you.
Did you have something good? Okay. Can we get an online question, please?
Okay. IID database is not up to date for inactive ingredients used in transdermal products. What is the target deadline to update IID?
Nobody's got that one? Understandable.
I think Dr. Ronnie will correct me if I'm wrong, but it will be updated case by case. But if you think it's not updated, you can send us control correspondence and ask us about it.
Okay. Next online question, and again, if you have questions here in the room, please jot them down. Come on up to the microphones. But let's take our next online question, please.
All right. Is it allowable to develop a TDS product where the residual drug amount would be higher than the RLD but lower compared to other approved TDS products?
I'm not sure if there are any cases where that exists. Are there? There are? Oh, okay. Sam is in the back saying yes, there are. Maybe.
Sam, yeah, if
We
you
can
want to
have Sam.
grab the microphone there. Or, oh, okay. Or
I guess we have run across situations where the RLD product was discontinued and it had a higher drug load compared to the current reference standard. So I guess the question is it acceptable to develop a product that is lower than maybe the RLD but not higher compared to the current reference standard? And I think as Bob referred to in his presentation, the residual drug guidance recommends this, and we also recommend designing a product that is based on our currently available technology and minimizing the drug load as much as possible, because it does have safety implications in certain instances.
alrighty. And again, if there's any questions at the room, please come up to the microphone. Can we get our next online question, please?
Does FDA always expect to see IVPT studies submitted in the ANDA comparing heat effects with the test and reference TDS product? Also, how large do these studies need to be?
Yes, we expect to see IVPT studies, and you need to capture different heat effect scenarios, and depending on the variability of data, you may need multiple donors, but our recommendation is at least 4 donors and 4 replicates.
All right.
Do you want to add anything?
Do you have anything to add? Okay. Next online question, please.
To what extent does FDA expect clinical studies like the PK/BE study or comparative TDS adhesion studies to be performed under real-world conditions?
So I guess the question is, how do FDA expect to see the TDS that was conducted in the comparative PK/BE study or the comparative adhesion study to be conducted in the real world situation? Is that right? So I think currently what we recommended is what in the product specific guidance and the general adhesion guidance. Right now, we haven't recommended that, but we are thinking about that in the consideration.
So I'll also add to that to say that the main concern is the product going to behave the same for the patient? So if there's an obvious situation where the patient is going to use the generic product, for example, it's a 7-day wear product, and you expect them to bathe maybe daily, perhaps take a shower, what you wouldn't want to have happen is that the generic product, which will get substituted at the pharmacy, falls off on the first day in the shower. So if there's a real world situation like that, it shouldn't be a protocol parameter for the comparative adhesion study that exposure to water during the 7 days is prohibited. Now, those are the things that you want to take into account as you're developing your protocol designs.
All right, let's go with our next online question, please.
Can a topical patch with a lower drug load than the RDL be submitted in an ANDA, or would it need to be submitted as an NDA?
Topical delivery systems that we also refer to as topical patches, the strength is defined different than the transdermal delivery system. There, the strength is actually the drug load in the system. And if you have developing a system that has a different drug load than the RLD, then you cannot submit it as an ANDA because it's not pharmaceutical equivalent to the RLD.
And sorry, my understanding was it was a concentration basis for the strength, correct? Or,
Drug load and concentration.
drug load. Okay, I thought drug load could differ. Anyway, sorry.
It's concentration, yes.
A concentration base.
Yes.
So for instance, lidocaine topical delivery systems, they're all 5%. But the weight of the total system can differ, and the drug load can differ.
Okay, let's take one more online question, please.
What is the recommended clinical study to justify adhesive raw material specification range? Do the ANDA applicants need to do a clinical study on low and high end of adhesive raw material specification ranges?
If an applicant wishes to do those kind of development studies, that's great, but I've never seen an applicant do them, and it's not a requirement. The idea is that the applicant, in my presentation, I was suggesting that raw material suppliers for adhesives, they tend to have very wide acceptance limits because of the fact that those adhesive materials may be used by a wide variety of industries, and not just limited to the pharmaceutical industry. And so as a pharmaceutical company, you may need to impose your own tighter limits in order to ensure product quality and performance.
Okay, and we've got a couple minutes left before I wrap it up. I wanted to, based on the questions that you heard, does the panel or our subject matter experts in the back of the room have anything they want to add based on what was discussed? Going once. Anything in the back of the room? All right, please help me thank our panel for coming and answering.